This recording covers the chapter on muscle tissue, how muscles transform chemical energy into mechanical energy, and is part 1 of 2. In looking at the histology of muscle tissue, we notice that skeletal muscle tissue is comprised of long cylindrical cells that we refer to as muscle fibers. You'll notice that there are lines that run perpendicular to the length of the skeletal muscle fibers. These lines are referred to as striations. Striations appear because of a very regular pattern of actin and myosin protein that is found within skeletal muscle fibers. Skeletal muscle tissue forms when hundreds of myoblasts fuse together to form a long cylindrical muscle fiber. When the myoblasts fuse together, their nuclei tend to get pushed to the periphery or the edge of the muscle fiber. So we see multinucleated skeletal muscle fibers. Skeletal muscle tissue is voluntary. A thought is required for contraction to occur. If I generate the thought to raise my hand, there are muscles that will contract in order for this motion to occur. In looking at cardiac muscle tissue, we also see striations running perpendicular to the length of cardiac muscle cells, which tend to branch out. Cardiac muscle tissue has 1 to 2 centrally located nuclei and is referred to as being involuntary, which is a good thing. We don't have to think about our heart beating. The heart consists of 4 chambers. There are 2 upper chambers, which are called the atria, and 2 lower chambers, which are called the ventricles. The heart is designed to beat in sync so that the atria beat together, there's a pause, then the ventricles will beat together. Therefore, the heart beats atria, ventricles, pause, atria, ventricles, pause, atria, ventricles, pause. There's another structure that's not listed on this slide that is important. You can notice in the picture that there are some darker lines which are representative of intercalated discs. Intercalated discs are structures found between cardiac muscle cells where we have desmosomes and gap junctions. Gap junctions allow ions to flow from one cardiac muscle cell to the next in order that the heart can beat in sync. Intercalated discs also have desmosomes. Desmosomes are those spot welds that hold those cells tightly. That means when the heart does contract, the muscle tissue won't tear apart from itself as it's beating in sync. Lastly, we have smooth muscle tissue, comprised of cells that have a spindle shape, which means the ends taper off into little V-like structures. Smooth muscle cells are not striated because they don't have that same regular pattern of actin and myosin within them. When they contract in a ringing fashion. Smooth muscle cells have 1 centrally located nucleus and are also involuntary. We find smooth muscle cells in the walls of hollow organs, such as the urinary bladder, the intestines, the stomach. Again, smooth muscle is involuntary, which is helpful because if you're digesting the sandwich that you ate earlier in the day, you don't have to voluntarily think, squeeze, shorten, squeeze, shorten, squeeze, shorten, to move food through your digestive tract. There are certain characteristics that are unique to muscle tissue. First of all, it is excitable. It can carry an action potential, which is an electrical charge. This electrical charge can be generated and transmitted along a cell membrane. Muscle tissue also has the characteristic of contractility. It can contract or shorten when stimulated by action potentials. Remember, when you hear the word action potential, think of an electrical charge. Muscle tissue can also stretch or extend, which is the characteristic of being extensible or having extensibility. Muscle tissue is also elastic, which means that it is able to return to its original length and shape. Muscle tissue has many functions. We know that it helps produce body movements. That means that there's a concerted effort of coordination between our muscles, our joints, and our bones. Our muscles help to stabilize our body positions so that we can move more efficiently. This also includes maintaining posture and joint position. We can move and store substances within our body. There are special circular muscles known as sphincter muscles that can keep substances in certain areas of our body before releasing them to others. For example, there are internal and external sphincters associated with the urethra, so urine doesn't constantly dribble out of your body. There are internal and external sphincters associated with the anus. There are sphincter muscles inside the digestive system to hold food in a certain area so it can mix with other substances such as hydrochloric acid within the stomach. We also have a sphincter muscle around the eye, which allows us to close our eyes. That's orbicularis oculi, as well as a sphincter muscle around our mouth, orbicularis oris. Cardiac muscle helps to move blood through our bodies. Smooth muscle helps to move blood through our bodies, urine through the body, as well as food through the body and nutrients. Our skeletal muscle is important because it helps to move venous blood back to the heart as well as circulate lymphatic fluid through our bodies. Our muscle tissue also helps us generate heat. If you've ever stood outside on a cold day, shivering, that generation of heat by the contraction of skeletal muscle will help you maintain homeostasis. If we were to build a skeletal muscle, we would start with a single skeletal muscle fiber. You can see this skeletal muscle fiber at the bottom of your picture. You'll notice the term sarcolemma. The sarcolemma is the membrane that wraps around that single skeletal muscle fiber. Imagine this pipe cleaner is one skeletal muscle fiber that was formed by hundreds of myoblasts that fuse together. It has a sarcolemma membrane wrapping around it. Around each skeletal muscle fiber is dense irregular connective tissue called endomysium. Endo means inside. In order to build a muscle, we need more than one skeletal muscle fiber. So we're going to get a bunch of them together. Now we have created a group of skeletal muscle fibers that makes up a muscle fascicle. In order to hold this muscle fascicle together, we need another layer of dense irregular connective tissue known as perimysium. Peri means around. And then lastly, in order to make a muscle, we need groups of fascicles bundled together to make a skeletal muscle. Imagine this is biceps brachii. And around the skeletal muscle is dense irregular connective tissue that we refer to as epimysium. Epimysium contains the prefix epi, which means over or above. Therefore, the epimysium is the connective tissue above all of this skeletal muscle. All of the connective tissue layers inside, around each muscle fiber, around the fascicles, and on the outside blends together through the length of the muscle to form a connection at the end of the muscle. Whether it is a tendon or an aponeurosis, it's actually formed by all three layers of dense irregular connective tissue that, as they blend together, have a more regular pattern of collagen fibers. That is why tendons and aponeuroses are dense regular connective tissue. Each individual skeletal muscle in your body is considered an organ supplied by a nerve, an artery, and a vein. It's very common to see arteries, veins, and nerves running together in what is referred to as a neurovascular bundle. Skeletal muscles have to have a rich blood supply so that their mitochondria can be fed with oxygen and nutrients to produce adenosine triphosphate, which you remember is the cell's chemical form of energy. As I mentioned previously, skeletal muscle fibers arise from the fusion of over 100 myoblasts during embryonic development. Therefore, mature skeletal muscle fibers will have over 100 peripherally located nuclei. The mature skeletal muscle fibers cannot undergo mitosis. All the fibers that you have are present at birth, and the cells last a lifetime. Therefore, skeletal muscle does not regenerate well. Here we have a picture of a muscle fiber. Sometimes muscle fibers are referred to as myofibers because the term myo means muscle. You can also see the sarcolemma, which is the special term for the plasma membrane around the muscle fiber or myofiber. Inside other cells, we have a plasma membrane, and the inner contents between the plasma membrane and the nucleus are referred to as the cytoplasm. With muscle fibers, we call it the sarcoplasm. By the way, the term sarco means flesh. So the sarcoplasm is the inner contents of a skeletal muscle fiber. You'll notice that the skeletal muscle fiber or myofiber contains cylinders referred to as myofibrils. be very careful because it's easy to get confused with regard to the level of structure that you're viewing. So pay very close attention. The innermost cylinders are the myofibrils that make up a muscle fiber. These myofibrils are actually contractile organelles, and it is within these contractile organelles that we see a very specific organization of proteins known as actin and myosin that create the striations. So myofibrils are where contraction occurs, and they also are responsible for the creation of the appearance of striations. 80% of the volume of a muscle fiber is made up of myofibrils. We also see inside the sarcoplasm lots of mitochondria. The term mitochondria is plural for the singular term mitochondrion. Within the myofibrils, we have myofilaments. These are the special proteins that make up the myofibrils and are responsible for contraction. Within the myofibril, we have thick protein filaments called myosin and thin protein filaments called actin. The myosin is identified in a kind of purple color on this picture. The thin actin filaments are designated by a green line. You'll notice that because of the very specific arrangement of actin and myosin proteins, we see this pattern of bands that form. We have at the very bottom of your screen, you'll notice a dark A band where we have an overlap of actin and myosin only at certain portions within that dark A band. On either side of the dark A band, we have light I bands because the appearance is lighter. And you'll notice that the way the actin joins together, we have a zigzag appearance forming the Z disc that goes right in the middle of the light I band. Pay attention to the things that you find in these areas. In the I band, the only thing we find in terms of proteins that you need to know is actin. In the dark A band, there is yet another area that is formed called the H zone. You'll notice that actin does not exist in the H zone. There's only the dark, thick myosin in the H zone. In the H zone, and something that's not shown on this picture, is a middle area referred to as the M line. So right here in the middle, that is the M line. The M line is in the middle of the A band. The actin and myosin are so arranged that the filaments can slide inward towards the H zone for contraction to occur. There's something else you need to know on this picture. And I just realized the M line is identified down here. There is a structure produced by an organelle called the sarcoplasmic reticulum. The sarcoplasmic reticulum surrounds the myofibrils and forms a network of smooth endoplasmic reticulum that is important for storing calcium. You'll also notice on this picture the term transverse tubules. Transverse tubules are invaginations of the membrane that surrounds the muscle fiber. In other words, the sarcolemma is going to dive down into the sarcoplasm to carry electric impulses and communicates with the sarcoplasmic reticulum to allow calcium to flow into the sarcoplasm. We're going to take a look at this again and again to help you understand this process. When we look at the proteins involved in contraction, we see myosin, which is the thick filament. In order to form myosin, approximately 300 myosin molecules are bundled together with their tails forming the central portion of the thick molecule, and the little myosin heads, which look like little golf clubs, are facing outward at the end of each molecule. The thin filaments are composed of a protein known as actin. Now, actin actually includes two parts. There is a filamentous, which means it's like a filament or a string, filamentous portion or F-actin, and then there's a globular portion, which is what these little yellow beads are that you see here. Those are called G-actin subunits. Now, I'm not telling you this because I want you to remember the little pieces parts of actin. However, what is important is that the thin filaments of actin are where we find myosin binding sites. So the myosin heads will bind to the myosin binding site on actin. You can see half of a little dark circle on the actin subunit. See the little dark area right there? That's covered up in this picture. The dark part is covered up by another protein called tropomyosin. So we have myosin as our thick filament with its tail and its myosin heads. The myosin heads eventually will bind to myosin binding sites on actin. When the myosin heads bind to myosin binding sites on actin, they form a structure that is called a cross bridge. When myosin and actin are attached, they form a cross bridge. Remember that myosin attaches to myosin binding sites on actin. I keep saying that because that's important. The actin, as you can see, is shaped like a little twisted string of beads. And what regulates actin and myosin to determine whether or not cross bridges can form are regulatory proteins known as troponin and tropomyosin. Let's take a closer look at troponin and tropomyosin. Tropomyosin is this brown protein strand that covers over the myosin binding sites found on actin. When calcium is released by the sarcoplasmic reticulum into the sarcoplasm, calcium will bind to troponin. Remember, when molecules bind to one another, it changes their shape. So when calcium binds to troponin, it changes the shape of troponin. Troponin is bound to tropomyosin. So when troponin changes shape, it changes the shape of tropomyosin and slides it away to remove the tropomyosin from the binding sites, allowing them to interact with myosin. To recap the things that are involved in a skeletal muscle fiber, remember that the skeletal muscle fiber has a membrane called the sarcolemma. The sarcolemma will invaginate or dip down inward, forming a structure called the transverse tubule. The transverse tubule will communicate with the sarcoplasmic reticulum, which normally stores calcium. We know that inside the skeletal muscle fiber is the sarcoplasm, 80% of which is made up by myofibrils. These myofibrils have protein filaments inside them arranged in a very specific pattern. The pattern, by the way, forms the contractile unit that is the functional contracting unit of a myofibril called the sarcomere. This is the first time I'm introducing the term sarcomere to you. A sarcomere is the functional contracting unit of each myofibril, and there are thousands of sarcomeres along the length of myofibrils, depending on the length of the myofibril within the skeletal muscle fiber. The sarcomere is defined as being the area from one Z-disc to another Z-disc. Remember that we have dark A bands, light I bands, an H zone in the middle, and an M line in the middle of each sarcomere. The myofibrils are attached to the sarcolemma by a protein called dystrophin. If dystrophin is not made correctly, that could lead to problems known as muscular dystrophy. We also see lots of mitochondria in the sarcoplasm. We find myoglobin inside the sarcoplasm. Myoglobin stores oxygen in skeletal muscle fibers, just like hemoglobin carries oxygen in our bloodstream. We also see glycogen granules inside the sarcoplasm. Remember, glycogen can be broken down into glucose to be converted into adenosine triphosphate. And we store glycogen in our skeletal muscle fibers and the liver. When you read through your text, you'll read about other proteins involved in the sarcomere, such as titin, which you can actually see on here. And I'm not going to ask you about the titin filament. It actually does help kind of anchor structures together within the sarcomere so they don't get pulled apart, and allows for recoil because the titin filament actually has elasticity in it. It is an elastic filament. However, for our purposes, you need to know about the actin filament, the myosin filament, and the structures of the sarcomere, the dark A band, the light I band, the H zone, the M line, the Z disc. Remember that a sarcomere is defined as being from Z disc to Z disc. Everything in between, from Z disc to Z disc, is the sarcomere. The sarcomere is the functional contracting unit of the myofibril. There are thousands of sarcomeres along the length of each myofibril. Okay, here's more about the sarcomere. We've got the dark A band is the entire length of the myosin. The I band only contains actin. The H zone only contains myosin. The Z line forms as an anchor site for actin. The M line is the middle of the sarcomere. And myofilaments do not shorten. They move inward towards the M line. They slide past one another. So striations disappear and the H zone gets smaller when contraction occurs. There is a good YouTube video on here that you can't watch on this because it's recorded. I'll see if I can get that to you guys in one of the video banks that we have for all of this information. Or you can write that down. Again, during contraction, what happens to the sarcomere? Basically, the A band is going to stay the same length. However, the I band will shorten and the H zone will shorten. Let's take a closer look at the events that occur at the motor end plate between the neuron and the skeletal muscle fiber. Just so you know, I do have a different video in Blackboard where I use different pictures from another source. So you will also want to look for the video on excitation-contraction coupling. I'm going to explain it as best as I can here with these pictures. However, I really believe that the other video may help you as well, so please make sure that you watch that. When we look at the events of innervation and excitation, remember that a nerve impulse has to travel to a skeletal muscle fiber in order for contraction to occur. A nerve impulse is often referred to as an action potential. It's simply an electrical charge. The action potential travels from the neuron cell body away from the cell body towards an effector, which is a muscle or a gland. In this case, we're looking at a skeletal muscle fiber acting as the effector. The axon carries the impulse, and as the axon reaches its end, it splits into branches. At the end of the branches, the axon tends to swell into a structure referred to as a synaptic end bulb. You may also see the term synaptic end knob or axon terminal. They're all the same thing. Because of the electricity that's traveling down to the synaptic end bulb, calcium voltage-gated channels will open up, allowing the influx of calcium into the synaptic end bulb or axon terminal. As calcium moves into the synaptic end bulb, it will trigger synaptic vesicles to merge their membranes with the membrane of the synaptic end bulb. These synaptic vesicles contain a neurotransmitter called acetylcholine. When the synaptic vesicles merge their membranes with the synaptic end knob membrane or synaptic end bulb membrane, acetylcholine is released into the synaptic cleft. Acetylcholine then binds to acetylcholine receptors on the folded area of the sarcolemma of the skeletal muscle fiber that you already know is the motor end plate. When acetylcholine binds to the acetylcholine receptors, that opens up those receptors to act as channels that allow sodium to come in. When sodium enters in to the skeletal muscle fiber, that causes a change on the voltage of the membrane of the sarcolemma. This local change on the membrane of electricity or voltage on the sarcolemma spreads across the sarcolemma. And as this change in voltage spreads across the sarcolemma, it is also carried into the sarcoplasm by way of the transverse tubules. Remember that the transverse tubules are simply invaginations of the sarcolemma. The transverse tubules wrap around each myofibril inside the skeletal muscle myofiber. What you can't see on this picture is that there are proteins embedded in the membrane of the transverse tubule that communicate with other proteins embedded in the terminal cisternae of the sarcoplasmic reticulum. When the voltage travels down, that end plate potential, that local change in voltage travels on the sarcolemma and through the transverse tubules. That change in voltage changes the proteins embedded in the transverse tubule, which affects the shape of the protein embedded in the terminal cisternae. When the shape of these proteins change, the shape change of the protein embedded in the terminal cisternae allows the release of calcium ions from the sarcoplasmic reticulum into the sarcoplasm. So now calcium gets released into the sarcoplasm. Calcium is stored in the sarcoplasmic reticulum. When the sarcolemma of the skeletal muscle fiber depolarizes because of that end plate potential that occurred with nerve stimulation and the release of acetylcholine, as the electricity spreads from that end plate potential across the sarcolemma and goes down the transverse tubules, remember that the proteins embedded in the transverse tubules will change shape in response to the effect of the electricity. Those proteins will mechanically change the shape of proteins embedded in the sarcoplasmic reticulum, allowing for calcium to be released into the sarcoplasm. In this picture on the right, you can see the release of calcium as those protein channels open up. Once calcium is released into the sarcoplasm, it will bind to troponin. Because troponin is bound to tropomyosin, when calcium binds to troponin, it changes the shape of troponin, thereby affecting the position of tropomyosin. Tropomyosin will move away from the myosin binding sites on actin, exposing them to the myosin heads. Myosin can now form a cross bridge with the actin subunits. This is what occurs when nerve stimulation happens. When nerve stimulation ends, the calcium has to be actively pumped back into the sarcoplasmic reticulum. That process also requires energy in the form of adenosine triphosphate. The contraction cycle, as with any cycle, because it exists in a circle, we could really look at a picture of what happens within the cycle at any point. We're going to start with this picture at the top of the picture, where we have adenosine triphosphate binding to myosin heads. When adenosine triphosphate binds to myosin heads, there are enzymes within the myosin head that will hydrolyze or split apart adenosine triphosphate into adenosine diphosphate and inorganic phosphate. Remember that breaking bonds releases energy to do work. The breaking of adenosine triphosphate into adenosine diphosphate and inorganic phosphate gives the myosin head the energy to come into its cocked position in order that it may have the proper position to potentially bind to actin. Once the myosin binding sites on actin are revealed, which happens when calcium is released, which binds to troponin, which pulls tropomyosin off of those sites, the myosin head can now bind to that myosin binding site on actin, forming what's known as a cross bridge. When a cross bridge occurs, the inorganic phosphate is released, further strengthening the bond between actin and myosin. At this point, the adenosine diphosphate will be released, and the energy from the release of the adenosine diphosphate will give the myosin head the energy for a power stroke, where the cross bridge rotates. And this kind of occurs, it's release and then pull inward. So the force is generated inward, and the myosin heads are literally pulling the actin inward towards the M line, towards the center of the sarcomere. We are now at this part down here. We've had the end of the power stroke occur. The only way that more power strokes can occur is to detach the actin and the myosin. These are still bound together. Detachment of cross bridges can only occur when we have fresh adenosine triphosphate come in and bind to the myosin head. That causes the myosin head to release from the actin, and then the myosin head will also hydrolyze the adenosine triphosphate, bringing it into adenosine diphosphate and inorganic phosphate, and then we're back to the original place where we started. And this is a contraction cycle. So we have to have fresh adenosine triphosphate in order for the bond between actin and myosin to occur. If we don't have fresh adenosine triphosphate, well, there will be no release between the actin and the myosin. And that's usually what we see when people are not producing enough adenosine triphosphate. When does that happen? Well, when people die, approximately 4 hours after they have deceased, we notice peak rigidity in muscles at about 12 hours, and this will gradually go away over the next 48 to 60 hours. So we'll start to see rigidity or rigor mortis 4 hours after someone has deceased, and again that can remain anywhere between 48 to 60 hours. That's how forensic scientists can tell how long a person has been deceased. It's usually the level of rigor mortis within the body. Looking at the cycle of contraction, it will repeat as long as adenosine triphosphate is available and calcium levels are high. The myosin heads will continue to rotate back and forth in unison with one another so that they're always generating some force as others are detaching. And the sarcomere is capable of shortening to half of its resting length. Again, I've got some really good videos in Blackboard that are not just the ones I made. There are also some other good videos that you should check out, because you can see the animation of the myosin heads moving the actin inward towards the M line of the sarcomere. So please make sure you're checking out those videos on Blackboard. When contraction occurs, remember we first have to have a nerve impulse. So we've got an action potential arriving at the neuromuscular junction, releasing acetylcholine, which binds to its receptors, opening sodium ion channels that lead to an action potential, which we call an end plate potential, because it happens at the motor end plate on the sarcolemma. That action potential or end plate potential will travel along the transverse tubules. They have protein channels embedded in them, which are going to respond to the change in electricity, because proteins are affected by electricity. So when their shape changes, it also changes the shape of proteins with which they communicate embedded in the sarcoplasmic reticulum in the terminal cisternae. That opens up calcium channels to release calcium into the sarcoplasm. Calcium binds to troponin, that pulls tropomyosin out of the way, and now the myosin heads can form a cross bridge with the myosin binding sites on actin. And then we have what is referred to as the sliding filament theory, where the actin is pulled inward towards the middle of the sarcomere by the myosin heads. The muscle shortens and produces tension. Now, suppose there's no more muscle stimulation, then relaxation will occur. And what happens with relaxation is the acetylcholine that was bound to the acetylcholine receptors will move away from the receptors by way of diffusion. There's also an enzyme called acetylcholinesterase that will break down acetylcholine into its components of acetyl and choline, which will be taken up actively into the synaptic end bulb to be reconfigured and repackaged as acetylcholine in the synaptic end knob, so they can actually be recycled. Calcium is also going to be actively pumped back into the sarcoplasmic reticulum with relaxation. Remember, active transport requires energy, so ATP is required to get the calcium back into the sarcoplasmic reticulum. Remember, we had enough ATP to bind to the myosin head to break that bond between actin and myosin, and that allows the thickened thin filaments to relax. So lastly, the muscle will lengthen and relax. Okay, this is just another recap of what I just said. The sarcoplasmic reticulum has active calcium pumps that are going to use adenosine triphosphate to pump calcium back into the sarcoplasmic reticulum. What's kind of cool is the concentration of calcium when a muscle is at rest is 10,000 times higher than it is in the sarcoplasm. So you've got 10,000 times more calcium stored in the sarcoplasmic reticulum than you'd find in the sarcoplasm when the muscle is at rest. Calcium depletion will cause the tropomyosin to cover up the active sites on the actin molecule, and cross bridges can no longer form with relaxation. Here is another recap of the events of contraction and relaxation in a skeletal muscle fiber. Remember, we have a nerve impulse arriving at an axon terminal of a motor neuron, triggering the release of acetylcholine. Here they actually use the words axon terminal. As acetylcholine is released, it binds to acetylcholine receptors and causes a muscle action potential. Technically, it's an end plate potential. Either end plate potential or muscle action potential are fine. This electricity travels on the sarcolemma into the transverse tubules. It will change the shape of proteins in the transverse tubules, which will mechanically change the shape of proteins in the sarcoplasmic reticulum, which releases calcium into the sarcoplasm. Calcium binds to troponin, which moves tropomyosin, exposing myosin binding sites on actin. We have contraction occurring, and I'm not going to go through all of that on here. And then after all that occurs, active transport pumps calcium back into the sarcoplasmic reticulum. The troponin and tropomyosin go back into place and the muscle relaxes. So this is just a very small little picture of what occurs. Again, I've got some really good videos on Blackboard with some different pictures that I think you might find handy. The process of the thick and thin filaments sliding past one another is referred to as the sliding filament mechanism or sliding filament theory. It occurs when the myosin heads attach to the actin and walk it inwards toward the M line. This shortens the sarcomere as the Z discs come closer together, and the action results in shortening the muscle fiber, and thus the entire muscle becomes shorter. This picture is another recap of cross bridge formation. Remember that after calcium has been released into the sarcoplasm and binds to troponin, That pulls tropomyosin away from the myosin binding sites on actin. With an energized myosin head, a cross bridge can form as myosin binds to the myosin binding site on actin. How do I know this is energized? Well, I can see that the adenosine triphosphate has already been hydrolyzed or split apart with water into adenosine diphosphate and inorganic phosphate. That's what the little I stands for. When you see PI, it means inorganic phosphate. So it's a phosphate group. It does not have carbon and hydrogen, therefore it is inorganic. Once the cross bridge has formed, the inorganic phosphate will actually be released, strengthening the bond between actin and myosin. And adenosine diphosphate will be released next, causing the power stroke to occur. Most books don't do this as separate events. However, one of the videos that I have for you in Blackboard shows it really nicely, that the inorganic phosphate and the adenosine diphosphate are actually released separately. After the power stroke occurs, we need a molecule of fresh adenosine triphosphate to attach to the myosin head, causing the myosin head to detach from the myosin binding site on actin. With that fresh adenosine triphosphate, knowing that the myosin head has enzymes that will split it apart, we see that the myosin head is now energized because the bond has been broken, breaking adenosine triphosphate into adenosine diphosphate and inorganic phosphate. This cocks the myosin head, it moves it. The energy from breaking apart adenosine triphosphate moves the myosin head into its cocked position so that it can have another bond with the exposed myosin binding site on actin. So as long as intracellular calcium, the calcium levels, as long as the calcium levels within the sarcoplasm are high enough, that's going to keep the binding sites available. So this can repeat again and again and again as long as we have enough ATP and calcium to allow that to occur. That's the end of this part of the slideshow. I'm going to move on and go to the next part for this chapter.